Hello, this is Julian from Straight from a Scientist podcast. I'm here with a very interesting man, Miguel, who is studying nutritional neuroscience and specifically people suffering from mild cognitive impairment and how the nutritional advice can help them. So, nice to meet you, Miguel. Glad to have you on today.
Thank you. Thanks very much for having me.
So, tell me a little bit about your story. What got you into science? What got you into the nutritional neuroscience field?
Um, so, um, I'm a mature student. I'm in my 40s, so
Still young, still
I
young.
actually, yeah, no, good. But I'm not just a recent graduate in my early 20s. So in my early 20s, I actually studied business, and when it was actually trendy to study business, and took that road. But then I was always attracted to science, and the first contact with science was environmental science. So I went back to university in the UK. I'm Spanish, born in Madrid, but I I've lived in the UK for 25 years, so I've got the two passports now,
Oh
which is
wow.
quite cool. I like
Yeah,
I
that's
like
nice.
having British and the Spanish passport. So I went back to university and did environmental science, and I didn't quite finish the dissertation. I was going to do a master's on it, and then I decided to actually study nutrition because during the time that I was doing the environmental science, I got more and more attracted to nutrition matters, and I ended up doing a full first degree, so what in the UK is a BSc, so a bachelor's, a 4 year degree, bachelor's, a BSc honors in nutritional medicine. And that basically in the UK means the application of science to human health. So it's really translating science that you can apply in clinical practice with humans, as opposed to other kind of like nutrition degrees that don't really give you those kind of like clinical skills. So I really knew that I wanted to be working with people as opposed to working in a lab doing stuff with mice or with cells. So and as I started practicing, I got more and more attracted to the idea that you could do something with nutrients, with food particularly, but also with some supplements that when they are warranted, to help people who came to me and said, "I'm in my 40s now. I'm um, I'm struggling to remember things as well as I did when I was in my 20s. So I don't think I'm going to get Alzheimer's or anything like that, but I'm kind of, I haven't got that freshness of brain that I did in my 20s. And another situation was the ability to be creative and make decisions and kind of being in control of your brain as opposed to suffer from this kind of a foggy kind of brain that some people can get because of fatigue and so on. And
Yeah, I guess
particularly,
brain fog.
yeah,
I was going
sorry.
to say brain fog is a big problem nowadays, right? I mean, a lot of people just are experiencing brain fog, and it can be related to their diets and other things, right? I mean, we can get into that
Hmm.
later. Yep.
Yeah, definitely. So brain fog was something that I had heard of before, and I thought it was a bit of a myth, as who gets this brain fog thing, and I was working with a community of patients who had chronic fatigue at a clinic in London that specializes just in that area. So I was clinical director for a year there, getting the basis of what has now become a randomized controlled trial that's going on at the University of Surrey that they are doing in that area, a multiple intervention controlled trial using nutrition and mindfulness and breathing and things like that. So I was there just before the RCT was being or started, and I really got to understand how this thing is a reality, and sometimes it can actually precede other neurodegenerative diseases. So you've definitely, as you'll know, studying neuroscience yourself, there is the condition that is diagnosed, like let's say Alzheimer's, but by the time that is diagnosed, it may have been happening in your brain for about 30 years, perhaps more.
Exactly.
So you could get early signs, and you may not fully develop something that is a dementia type or an Alzheimer's type of condition, but you may be going towards that type of neurodegeneration that actually affects your memory and so on. And definitely chronic fatigue people, because particularly those who are stuck in a high cortisol kind of state for a long time, cortisol being such a catalytic kind of hormone, it really does break down the plasticity in the brain and the nervous system. So you can end up with a situation that many people who live on adrenaline and cortisol, like high-end executives and so on, experience, because they just break down. There's that point of no return for them where they really need to take time off. So there were a number of things really just coming into my life, telling me I needed to go back and do more brain studies. Really, I did some studying in the US with a medical organization, education organization called the American Academy of Anti-Aging Medicine. They do trainings for doctors and so on. Anti-aging, not as in Botox anti-aging, but as in
mental
nutrition
and instead of
anti-aging.
aesthetic anti-aging. Yeah.
Exactly, yes, and cardiovascular and so on. So I did a few modules with them, and I did this thing that they call a fellowship, where you then take an exam and become board certified. So I became board certified in anti-aging medicine, whatever that means. It doesn't mean anything in the UK, but it was kind of cool to do the exam and to kind of get more knowledge. And there was a lot of brain aging in there. There was obviously cardiovascular, brain, all sorts of things mixed in there. And that took me to think, okay, well, I think this is cool, but I really need something that takes me deeper into the pathways and how neurotransmitters are formed and what they do and what you can do in a clinical situation. So I took a while to kind of to make up my mind whether I wanted to do a behavioral type neuroscience master's or a cognitive neuroscience or a basic neuroscience and go back into the lab and actually spend time working with cells and mice and stuff like that. And in the end, I did a clinical neuroscience. I decided to go for something that was quite hands-on in the lab. So I'll tell you about my project that I worked on in a minute. But it was mainly with the idea that you come out and you have some tools to help people. So it was mainly a master's for individuals who are in some kind of clinical situation, so either a doctor or a nutritionist or a psychologist. So they have some tools to understand how to help somebody at the front end, at the clinical end, as opposed to at the basic end.
Yeah, I think that's
so that's
awesome.
how yeah.
That's really cool. I was just gonna say, the first time I remember when I got interested in, how food and things that you drink can affect your brain. I remember the first time I drank a coffee at age 13, I think, or 14. And I was so energetic for the rest of the day. And I was just thinking, I think in science class that day, I was like, how can something, just a small amount of liquid, change, so much, my mood, my activity, my, energy levels. So it's just amazing on the chemical level. I'm talking, I think nutritional neuroscience is a huge field that's kind of understudied. And I think a lot of things that we don't even realize are affecting, like you said, our memory and our cognition. So cool.
Yeah, sure. And that's what struck me really when I went back to college, and I thought I was going to be doing a whole new thing that was going to be hugely technical. And then I studied at the University of Roehampton, that has the reputation that the masters they've done, it's very it's got a long-standing history. It's approved by the Federation of Neuroscience Societies, Federation of European Neuroscience Societies, the FENS.
The FENS.
So it's
nice. Are you a member of the FENS?
Yeah,
Cool.
So it's kind of a very well respected degree, and it had a big component of going back to the lab, which I wanted to do because I had done a little bit of it, but I really wanted to understand. I thought I really wanted to get into a brain and actually understand what went on inside the brain.
Exactly. Cool.
Yeah, so I did that, and then I realized that a lot of the time I was speaking about nutrients, and I thought, wow, this is amazing. I'm talking about things that I already know, but I'm talking about them in a different way. So it was all about folic acid and
Exactly.
vitamin A and methyl donors and B12 and all of this kind of stuff. And I'm thinking, this is mind blowing for me because I knew there was going to be some kind of overlap, but in the end, the work that I had to write was mostly about nutrients. It was really great for me because I enjoyed looking at things from a different angle. But equally, that was what really then got me into looking at what I could do in the lab that wasn't completely alien to what I know already, and creating a bit of a bridge between my nutrition and neuroscience. So I looked at the effect of various nutrients on the mitochondrial transcription factors. So I looked at the effect of vitamin C, serine, and coenzyme Q10
Mm-hmm.
on the actual transcription of factors like NF-kappa B, NRF2, ARE, all of these kind of antioxidant, anti-inflammatory molecules that we talk about a lot, TNF-alpha, all of these kind of things. And that was very cool because I got into actually working with these things in my hands so I could understand how neural stem cells were actually growing or dying according to whatever growth factors I was giving them, what reactions they were experiencing, and then I got to see quite tangibly what happens when you give them even just a tenth of a microliter more of something. So
Yeah, it changes
so you're
everything.
dealing with tiny amounts that it's almost like a leap of faith. You need to believe that you have actually put some in the petri dish because you don't actually see it,
Wow.
and that was very different to working with humans and working at the clinical end. So it gave me a really good vision of how neuroscience actually is really hugely. It's got a huge breadth of application from that very basic kind of cell studies and animal studies to the clinical end. But being a clinical kind of person, I really wanted to go back and see things in the real world. So I completed my master's and I got a scholarship with Santander Bank and for my project that was quite rough in the proposal that I put to them at the time. It's developed into something a lot nicer now and more complex than I thought. But it's basically an understanding in the real world of people who go to clinics to talk to nutrition guys about memory issues, particularly borderline mild cognitive impairment kind of situation, what kind of nutrients they can get that actually work, and how you assess those nutrients so you can be more specific, or you can go with just food if you haven't got access to any assessment data, so you haven't got any idea as to whether you need more V12 or vitamin D or folate or anything. You just basically just give somebody some basic diet recommendations versus two other arms of a clinical study that I will be comparing. So diet only, bloods information. So you have information on vitamin D, vitamin B12, um, folic acid or folate in blood, C-reactive protein, a number of molecules that can actually help with plasticity in the brain. And then I'm going to compare what happens if you actually do a stool test and you get information from your microbiome. So I've got three situations there: diet only with no test at all, so basically just relying on symptoms, maybe a mini mental score
Yeah,
kind of
good idea.
or a yeah. And then I'm going to compare what happens when somebody actually gets information, gets advice that is more precise, so it's personalized to them based on nutrient levels in their blood, versus personalized to them because of various different configurations of bacteria that they could have, so different commensal bacteria or different metabolites in their stool.
Or even
So
parasitic bacteria, right? Deleting the ones or getting rid of the ones that are causing negative effects, right? I mean,
yeah, so a little bit like that. So how I got to the wanting to include that arm in there. So
that's a huge,
while I
huge thing. I think I did a small project on the microbiome and its connection to the brain. I guess the gut-brain axis. But they
Mhm.
actually in the paper they called it the microbiome gut-brain axis. So they added a whole
Yeah,
nother level,
yeah,
and it's
yeah. That
huge. You know, there's a lot of science coming out about it, and it's definitely an interesting topic.
Yeah, so this weekend, as soon as I've finished talking with you, I'm going to be spending the rest of the weekend writing a paper on that for the microorganisms journal, the MDPI journal,
Wow.
particularly on this subject. So how I came to want to include that is because I had had a lot of exposure to doing stool tests with people in private practice because I don't work in a hospital. I work in private practice. I have a network of doctors I work with. I get referrals from them. I get people coming to me directly because they find me and or word of mouth or whatever, and they come to me. And some of the tools that I use to assess their health are based on stool analysis. So I've been doing microbiome tests for the best part of 10 years,
Wow.
and they are quite sophisticated now in with regards to what they used to be 10 years ago. You can get a lot more information, and also the technology is a lot more precise. So you get the PCR technology, you can do 16S sequencing quite cheaply, and that gives you quite a precise view of what's going on at that given time. So I wanted to put together that kind of experience from the real world in a way that makes sense to other people. So my findings will help other clinicians as well to make better decisions. But also I spent a year working with Tim Spector, Professor Tim Spector at King's College, and he's one of the guys who's been doing a lot of the studies on the microbiome for the last probably 10 years because he's got a
Wow.
database of 14,000 twins. So he's a top cited scientist. I believe he's 1% top most cited scientist in Scopus. So he's cited like every day in a papers around the
Wow, that's
world.
amazing. Yeah.
And his thing for the last 10 years has been particularly focusing on the microbiome. So he's a professor of genetic epidemiology and twin studies. So he's looked at identical twins from birth until death, basically, and his database is about 14,000. So it's a very well respected data set, and it includes microbiome data. So I worked with him for a year in a project as an external consultant at King's College, and I got a really good insight as to how you can manipulate commensal bacteria, the things that you can do with basic things like eating more fiber or less fiber, or what type of fiber, or more fat, less fat. So I wanted to get an idea as to, from real world data, from tools that people like myself are using every day. So private tests that you can get on the internet or you can get via and diagnostic companies that serve the needs of private doctors and so on. What kind of data you're collecting? Because that's data that's the millions of tests done around the world every day, and they are not going anywhere. They are not written about in any papers at all. So I
Yeah.
wanted to see if I can actually put a methodology together to at least the community of users that I represent could have the means to feed back to a central database so we get more of an understanding of how the work that we're doing in private practice actually contributes to science and to clinical practice. So that's my
Yeah, and
bag
personal.
basically.
Yeah, I mean, there's a couple companies doing stool samples online. I think one is like Vis Biome or something. I don't know if you've heard of this. But the problem is, I think you receive the information and then, but you don't really know what it means. You know, you say, okay, I have
Yeah.
low lactobacillus, but what does that mean? Or I have high, streptococcus. So I think, yeah, that's really cool. Now, what I wanted to ask you, what are the main things that affect the microbiome? You said fiber has a has effect.
Yeah, so the main thing that affects your microbiome. So again, if you wanted to go to the endpoints in a way, even though I'm not doing a controlled trial, so to
And
speak, but
well,
if
just
I
based
would you
on your experience.
yeah, so if you wanted to measure endpoints such as better memory, energy, things that typically affect somebody who's got this spectrum of mild cognitive impairment and more towards Alzheimer's, but even before, even early, very early signs of neurodegeneration, because some of the very um, metabolites that you produce in the gut, like short chain fatty acids, like butyrate, for example, or butyric acid,
Yeah.
they travel systemically into the bloodstream and they cross the blood-brain barrier and they act as, let's say, as a general term, free radical quenchers. So that free radical formation that is contributing to the neurodegeneration is actually quenched to a certain extent by having better levels of or better concentration of butyric acid in the gut,
Mm-hmm.
because some of it is going to become systemic and is going to actually protect the brain and the central nervous system. Is going to travel via the vagus nerve to the brain, and it's going
Exactly.
to be absorbed via this gut-brain axis that people talk about. So the fact that you were saying before, um, this project that you were doing was referred to as the gut microbiome brain axis, so the microbiota, the gut microbiota brain axis. Actually, it makes sense to include those guys in there in that name because
Yeah.
if it wasn't because of them, if it wasn't because of the microbiota, you wouldn't be communicating probably. And these molecules are key to communication between the gut and the brain. So the acetic acid, uh, propionic acid, and butyric acid. You can measure very simply in a stool test, and it's one of the things that I do routinely in my clinic. And those are affected by fiber. So if you go from a typical American diet that's shorter in fiber than let's say a Mediterranean diet, where you're supposed to have more fiber from things like vegetables and fruit and whole grains and so on.
Yeah.
It's typically less sugary as well. So sugar, there's nothing wrong with sugar per se, but for bacteria, it's not great because foods that are very sugary, like a Krispy Kreme or something, it's going to hit it. It's going to make you feel very happy quite quickly because it's going to make you release some serotonin, and that's going to make you feel great for a few minutes. But then the reality is that most of the nutrients are going to be absorbed in the small intestine and they are not really going to be released into the colon so the bacteria can do anything with them. The fiber is too processed to get to the colon in any state that it can serve as a substrate for short chain fatty acids to be formed,
Mm-hmm.
compared to eating um, vegetables and fruit, for example, that are less processed. So your body needs to do the processing as opposed to some industrial process that has done that for you.
Yeah, and you get the
So
result of positive byproducts, like you said, like biorutate and all that. Interesting.
yeah,
Now, do
yeah. So that
you? Well, I had a quick question. Let's say you got your microbiota levels checked, and you saw that they were really low. What kind of steps, or what would you recommend to someone to kind of try to rejuvenate or repair their gut microbiota?
Yeah, so there could be various different strategies to do that. So normally I tend to stratify my results into three categories. So you're likely to get somebody who is just about average. In the UK, based on these studies that Tim Spector had done and published extensively, the average for the UK population was just around the 50%. So if you've got four quintiles, you're basically just around the middle. But in my experience, people who come to see me may not be the average people, i.e., they come to see me because they have some symptoms.
Yeah,
So they
exactly.
are typically outliers. So they typically people who, if they wouldn't fit into the average very well. So they tend to be a bit lower. So they tend to be anything from the top of the first quintile in a diversity score to the second quintile, but practically every time, and I'm talking like off the top of my head, but well into the 90% and plus, they would be below the 50%, below the average. And when that happens, you have somebody who has got low microbial diversity. You can have the average, which is the second stratum, and then you can have somebody who actually has got too much. So when you compare them with a healthy cohort, they actually have got too much bacteria, and that is not necessarily good, because bacterial overgrowth can then affect your small intestine. It can actually travel up into the small intestine. It can cause a lot of bloating, discomfort, and also the byproducts of protein metabolism and fat metabolism and fiber metabolism may be disrupted. They may become, the gut becomes what is known as dysbiotic. It becomes affected by imbalance, by a sustained imbalance. So if you have a low bacterial load, then that is the best possible situation. Because if you're in the middle, you're a bit boring. There's nothing much to kind of, you could do something, but it's the, when you want to say to somebody, your gut is very boring, it's very average, there's not much to say about it. You need to become a bit creative when you're explaining that, because otherwise it can sound a bit gray.
Yeah.
When you're in the low end of the spectrum, the best thing to do is to replace your own bacteria with live probiotics. So we met on Instagram, so you'll know that I Instagram about things like kombucha and kefir, and
Exactly.
so those are, for me,
The
I
best
used to use
options.
probiotics. Yeah, so I used to use probiotics in clinical practice. I used to give people, send them to some place where they could get probiotic capsules with, a few strains. When you compare a probiotic capsule, you may be spending $50 to $100 or like the equivalent amount of euros for something that's got anything from one strain. That normally is a bit of a gimmick, because you wouldn't just want one strain only in a ocean of
like you said, you want a
probiotics.
diversity, right?
Exactly, because that's what you're trying to do. So it doesn't make sense. You're trying to increase diversity. It doesn't make sense to give your body a super dose of one strain only. So when you go on to multi-strain, which is definitely the ones that have been shown in controlled trials to have a better effect on repopulation and inflammation markers and so on, you're getting into $50, $100 for a pot of probiotics that easily you could be spending that every month, and you could get like 10, 15, 20. Some of them have got 50 different strains, but and those would be very expensive if you want high dose and high diversity.
Yeah.
And when you look at things like kefir and kombucha, sauerkraut, kimchi.
Greek yogurt too. No, has
Greek
some.
yogurt, yeah. So Greek yogurt is great. The only thing is that you mainly have two. You have two types of bacteria in there. You've got some lactobacillus and some bifidobacteria,
Yeah.
which are fine. You've got two types in there, so you're putting two types. So my advice is to try and put in the diversity by introducing diversity from your diet. And first of all, introducing probiotic diversity. So you could get up to 30 different families of probiotics in a kefir, in milk kefir.
Wow, I didn't know that. Yeah.
Yeah, and if you drink water, kefir is reduced, because the actual organism, the symbiotic organism, is actually different. It looks a bit like crystals. The grains that make the combination of bacteria and yeast that make the water kefir actually look like crushed ice. And that produces about 5-6 different species of bacteria and yeasts as well. So there are some yeasts that are quite similar to the S. boulardii type of yeast that is probiotic, and the S. cerevisiae, which is the one that actually ferments beer. So those kind of probiotic yeasts are actually very useful when you need to repair your intestinal integrity, because they help you produce more secretory IgA, slightly raises secretory IgA slightly. So the local immunity of the gut actually improves quite dramatically. So that in itself is good. And then in kombucha, you've got a number of other things. So kombucha, not only does it give you the probiotics, so you've got a whole range of different bacterial and yeast species, and yeast as in good species. I mean, you can make bad kombucha if you mismanage it and you end up with an infection of something or another. But in general, it's quite difficult to break your kombucha brew because it's got so many natural preservatives. So it's got loads of acetic acid, which is why it can taste a bit vinegary.
Yeah.
It's got
Yeah.
it's got glucuronic acid, which is one of the main detoxifiers in the phase 2 detoxification in the liver when you're excreting xenobiotics and so on. You really need that. It's got enzymes, it's got short chain fatty acids. And the cool thing about the kombucha, which is what is one of the things that if I have the chance, I would actually like to study this in a little bit more detail, is how your body has got its own enzymes to break into polyphenols like the catechins in green tea, for example, but your own bacteria in your gut have got extra enzymes, and that's why the relationship with our bacteria becomes symbiotic, because your bacteria will have the key to break into a little door into the catechins of the green tea that you didn't have, and in that way you actually get a little bit extra out of that polyphenol. And the bacteria in the or the microorganisms in the kombucha actually have got the same ability. So when you make kombucha with tea, which is how it's made, so it's fermented tea, you're getting super complex polyphenols that no human organism would have been able to break down. And the long-term effects are unknown. They are likely to be good because it's been drunk for centuries in
Yeah, we
Japan
hope
and
so.
the Far East. Yeah, and it's known to be, in China, it was supposed to be the tea of longevity and all the rest of it. So we're guessing it's good. We don't know exactly because there hasn't been any actual controlled trials on what is going on with that. So the evidence is kind of based on traditional kind of medicine and so on. But I would love to know what's going on with those compounds, because they are recognized. They have been actually identified as being compounds derived from the original compound. They do the same thing if you do a second fermentation in your kombucha with pomegranate, for example. That's got it's a source of ellagic tannins, so ellagic acid. It breaks down those tannins into super complex tannins that have got a lot more ORAC ability, so much more antioxidant power. And it's not all about being antioxidant, because that's not really what we want them for. But polyphenols, actually, the latest thing about polyphenols is that they are prebiotics. So they help your microbes grow if they need to grow. So if you're on the low end of the spectrum, those polyphenols will improve your diversity and your numbers. And if you're on the high end of the spectrum, those polyphenols modulate the microbiome in a way that they are antimicrobial. So if you've got too much growth of Odoribacter or Prevotella or some of these that can become slightly inflammatory, and they can be associated, like the Prevotella, with things like Crohn's disease or MS and inflammatory conditions, they can actually modulate those down. They kill. It's almost like you do a cull of those that don't need to be there in such numbers. So they are really cool compounds, and they're really nice to drink as well. I make my own kombucha and I drink it every day, and I think it's amazing. So it's just introducing these things into the general public that I think is happening slowly.
Slowly, yeah. I was gonna say, you'll be happy to hear that this morning I had some kefir. And I also drank
cool.
I also brew my own kombucha. We had actually another podcast where we talked about the microbiome. And I did not know that the bacteria actually helps you to get better antioxidants and helps to do that. Wow, that's crazy. And and I didn't know about that about the pomegranate either, actually. One of my favorite ways to flavor it, because you are right, it tastes really vinegary, right? It's sour. The best way to flavor it is do the second ferment, which is by
second ferment. Yeah,
so good. You put some special, I normally put pomegranate, like you said, and I had no idea that it makes super antioxidants. So I'm going to continue to do that.
So this, exactly. So, and this is something that happens in the gut as well. So the kombucha is almost like a model of the gut in a way, when you're doing the fermentation, because the the same reactions are happening in the gut. So and that's why the tannins in pomegranate, following with the same example, they travel through the blood-brain barrier and they can actually do cool things in the brain. So they can protect against free radical formation and oxidative damage in not only in neurons but in glial cells and astrocytes, which are
Wow.
as important as the as the
It's the
neurons,
neurons,
and
yeah.
they act as, and the old school is that they are antioxidants. So yes, antioxidant, cool, it's fine, but the new way of thinking, and not so new, so it's been reported for probably for a good 10 years now, that the their ability to enhance communication, so their communication molecules, they really help the with neurotransmission, and they remove a little bit of the rust, a bit of the oxidation where it needs to be removed. But they seem to be, they work in a slightly different way to normal um, iron giving antioxidants, because they don't do that. They don't work like a vitamin C or like a vitamin A, for example, or a vitamin E. They work by, they are like drugs. They are xenobiotics. Technically, they actually are metabolized by phase 2 detoxification pathways in the liver, like real drugs, like paracetamol or ecstasy, so they basically, they need to go through the liver in the same way as a drug, and that makes them very unique compounds. So technically they could be bad for us if we took too many of them, which is
Hmm.
probably why, again, if we talked about supplements, I wouldn't go for a super duper mega dose of pomegranate whatever tannin in just one capsule, because you don't know what that's doing to you. That would be, for me, it would be silly to do that. Probably a bit less silly to do something that was a super dose of many different tannins together, because in nature they tend to come in a matrix of different compounds, so that's probably less damaging. But my understanding from literature is that little bits of those um, xenobiotics, which potentially could be dangerous in really high doses, they act in the body in a hormetic kind of way. So a little bit of them is good. If we had too many of them, then the curve actually goes into the detriment. It goes into the risk and harm, as opposed to the benefit, if you see what I mean.
Yeah, I feel. Now, I had a question. Now, all of this can be negatively affected by antibiotics. Have you done any research, or do you know anything about the effect of antibiotics on the gut microbiota?
Yeah, so not my own research, but when working with Team Specta, that was one of the things that came up all the time. So definitely, antibiotics do have a long-lasting effect. It depends on the type of antibiotic and the duration,
Yeah.
but a typical course of antibiotics can actually have an effect that can be measured years later. And it all depends, again, on how diverse your diet is. So if you have a diet that's the typical British or American diet that is not great with loads of colors, so not a lot of fresh fruit and vegetable, I'm talking about the worst possible American diet. There will be
The
guys out there who actually
McDonald's
eat
and
super
Burger King.
well.
Yeah.
Exactly, that kind of stuff. So if you have that kind of diet, technically, with some vegetables and stuff, but mostly potatoes and mostly, fried and processed stuff, and then you get wiped out because you've taken a course of antibiotics, then to recover that colonization of your gut, you're going to need to feed your own bacteria, as well as to put some extra bacteria. So whatever your colon gets during those days following the wipeout is going to start colonizing. And some of these bacteria you're going to get from foods, you're going to get from exposure, they are airborne. So whatever lands in your gut actually starts colonizing. So you could get into a situation that not only your gut is worse than it was before, it gets a different type of colonies in there as well.
Wow.
So it could actually change in nature. So to avoid that and to start putting the seeds and the fertilizer to start growing your lawn again. So think about it as a garden. So your garden has gone completely wiped out because you put too much Roundup or something, and it's just killed not only the weeds, but you've also killed the grass as well. So it's gone completely bleeds. If you want to recover it, you will need to lay down some new grass and grass seeds, and you'll need to feed the grass seeds so they grow. The grass seeds will be the new probiotics, and you get those from your yogurt and your cheese, if you can eat unpasteurized cheese as well. I know in the
Kombucha,
States
of
it's
course.
not, yeah, in the States it's not possible, but in many places in Europe, I mean in France, for example, is and in
Ooh,
Spain and
the
in the
raclette de
UK.
France is so good. Have
Exactly. So
yeah.
anything that's got a bit of yeasty kind of rind, or anything that has been made with unpasteurized milk, like in Spain, like manchego is made with
Ooh,
unpasteurized
manchego is
milk.
amazing. Yeah.
So you get nice bacteria from that, and you get some nice lactobacillus and bifidobacteria, and some other bacteria that not only produce lactic acid, they also produce propionic acid. So, for example, in Emmental and Gruyere, the hulls are produced by bacteria that are producing propionic acid. So those bubbles of the gas that is produced by the bacteria are the holes in the cheese. So you
Whoa.
get some nice bacteria in your gut from the cheese, and then yogurt, kombucha, kefir, sauerkraut, whether you make it yourself or you buy it in the shop. But if you buy it in the shop, just have a look that it says unpasteurized. So it's
Unpasteurized.
raw.
Yeah.
Yeah, so you get the bacteria in there. You still get a little bacteria in the one that's been pasteurized because it starts colonizing the same substrate, but you get a lot less. So, and ideally make it at home. It's dead easy. You just chop a few carrots and cabbage and put it in salty water with a bit of vinegar, and it will just grow its own bacteria 2 to 3 days later. So it's very, very easy. Things like tea as well, even just regular green tea, because it has been fermented for a few days before it's dried out. That fermentation actually carries some bacteria. So you're exposing your gut to nice seeds, and then you have the fibers, so nice things like onions, leeks, spring onions, so scallions. Um, those are great. Any sources of that, the type of fiber that those bulbs actually have, as well as brassica, things like the broccoli and the cauliflower and cabbage and kale and all of those things that are really, really good for the gut because they have a lot of fiber. Lentils, chickpeas, beans, all of those. And you can even put something like resistant starch, which is starch in things like potatoes and rice. So if you like your potatoes and your rice, if you cook them and you let them cool down, and you have a potato salad that has boiled potatoes that have gone cold, then you're doing two things. You get less calories out of the carbohydrate, which is cool if you want to keep your weight in check. And also you get more resistant starch, which is a super starch that your bacteria cannot break down properly. And in doing that, in trying to break it down, they produce more short chain fatty acids.
Wow. And they
So
multiply, right? I have heard that in order to, like one way, like you said, to feed the microbiome is to do exactly that. Boil the potatoes, let them cool down, and then eat some
yeah,
potatoes. Yeah.
yeah, and that's because at least half of that, if they've cooled down properly, it's going to turn into resistant starch. So resistant starch is almost like, imagine starch is these molecules of sugar that go into the small intestine, and they haven't been broken down like normal starch. That would be a bit like water or baby food when it comes to the colon. Resistant starch is almost like footballs, and then the footballs get into the colon, and your bacteria are playing football, playing soccer with the balls, and getting all excited, and having a good game of soccer. And then in doing that, they are sweating, and this sweat that they are producing is the short chain fatty acids, and the gut takes that via the enterocytes into the innervation. The tiny little nerves go into the vagus nerve. That's the broadband cable that goes right into the brain, and they travel to the particularly butyrate. There's loads of literature on how the butyrate actually travels to the brain, and it does a wonderful amount of things in there.
Wow. You just did an excellent you explained it excellently. Thank you. I wanted to ask you a couple things specifically about diets, and diet is a huge buzzword today. Everybody's trying a different diet. Some people are talking about the ketogenic diet. Other people are talking about fasting. And what are your thoughts on that? Let's get started with fasting. For me, I like fasting. Sometimes I fast until dinner time, about
Mm-hmm.
an 18-20 hour fast. And I have to admit, I feel great. And I feel like it rejuvenates me, gives me some energy. And what are your thoughts on that? What is your experience with that?
Yeah, so fasting does a number of things that have been very well reported in literature as well. So in animal research, which obviously, there's a lot fewer ethical constraints, is one of the ways that you diminish free radical formation in the brain. So when you're talking about the brain, particularly, you're actually diminishing that free radical attack to the brain tissue. So it's really good for neuroprotection. So it's a neuroprotective technique, really doing fasting,
Wow.
and it also does something very good for the microbiome. So your bacteria, when you're eating all the time, they are happy because they are being fed. So you have different species of bacteria in your gut, different all those 24 different commensal bacteria families. Each of those will like something slightly different. So some of them will like carrots, and some of them will like leeks, and the other one will like bread. And so if you're feeding yourself little bits and pieces of a variety of foods, you'll keep them all happy. When you're not eating, i.e. during the night, then what the bacteria do are then basically they go into a mode that they start chomping away at the lining of the bowel. So the lining of the bowel is made out of polysaccharides. So it's a combination of a little bit of protein, because there will be some secretory IgA and various immune proteins in there, but the mucus covering of the gut, almost like a little membrane on top of the actual enterocytes, that the cells lining the gut will be made of carbohydrates, basically. So while you're fasting, your bacteria actually eat that food, and that in itself is very healthy, because it stops you from accumulating too much of that mucus lining, which can then turn into a biofilm and attract pathogenic bacteria. And so it's very regulating. It regulates the two types of immunity as well, the Th1 and the Th2 immunity. So fasting is a fantastic thing to do. It's rejuvenating for animals. If you look at the effect it has on longevity, on things, anything from a fruit fly to a worm to a rat, is fascinating, the effect. Obviously, doing in a human is more ethically tricky. So there hasn't been that many studies done in humans on fasting or caloric restriction, which is kind of like the same thing.
Yeah.
But it's definitely one that I would advocate for. I also do fasting a lot.
Really?
I will definitely, yeah, I'll do a 16-hour fast probably 2 or 3 days a week.
Wow. Yeah. I skip breakfast almost every day. And I
Mm-hmm.
probably eat here in Spain. We eat really late, as you know, around 2 or
Yeah,
3 o'clock. So that usually gives me about a 16-hour fast almost daily. And I get my best work done in the morning. I just have a
yeah,
coffee, and I just I can really focus and get a lot of work done. And normally after I eat, I need to relax a little bit, take the siesta, or disconnect.
yeah,
So cool.
yeah,
Yeah, and occasionally
yeah.
I try to do a longer fast, but it's a little bit more difficult depending, when I try to get 18 or 20 hours, it can be a little bit difficult.
Yeah, yeah. And a little bit more constant is probably sending the right messages to your brain that are, again, it's just looking at, how we developed as humans. And if you look at anthropology and, going back to hunter-gatherers, and we had this constant exposure to fasting that was forced on us, because some days we had a lot of food, particularly in the winter months. We killed an animal and we feasted on that whatever zebra or whatever we killed, and we ate for 2 or 3 days non-stop because we were really hungry. And then we got some berries and some tubers or something, and we had a really big meal. But we could go for days where there was no food available to us as well. And so our bodies really is really used to that, because we still, like hunter-gatherers, living in a sci-fi world in a way. Our DNA hasn't really changed that much from hunter-gatherer kind of times. So we are that kind of a Paleolithic man and woman living in this world that's kind of science fiction, and food is available all the time. So taking your body back. There's this Walter Longo guy who's a scientist he's been publishing for many years about caloric restriction, as in the kind of like basic science, doing that with animals and cells in a lab. And he's just come out with a book for the general public on the anti-aging diet, or eating well for longevity, or something like that. Some things may be a bit more gimmicky, but what he talks about actually makes sense. And fasting and caloric restriction is one of those. I totally advocate that. That is the if that agrees with you, obviously it needs to fit into your lifestyle, and you need to feel well. And don't do it if you feel lightheaded. I mean, it's not a competition. Some people get very obsessed with, oh, you've done 20 hours, I need to do 22 hours. So, I can do more. I can do 2 days on water only. It's not a contest. You need to make it work for you. And even people who cannot do fasting for that amount of time because it doesn't work for them, just because they really feel they need the food. So it may be more like a behavior thing, but some of them may feel a bit shaky or something if their blood sugar is not very stable. Just not eating in between meals, that's another way of actually minimizing the calories going in and giving your body a rest. It's about economics as well in your ATP consumption in your body. And if you're always using the pathways that use the energy and the calories, like the mTOR pathway, that's very key to nutrient metabolism. And if you're always engaging those pathways, you're always using a lot of that energy that is needed to metabolize food. So if you give yourself a rest from food, from breakfast to lunch, and then from lunch to dinner, that's, instead of actually always eating snacks and stuff. So that's another way of doing it.
Yeah, interesting. Yeah, I think also it takes time. I remember when I first started fasting, and I did feel lightheaded, like you said, and I struggled a little bit, and my blood sugar got really low. But as I got used to it, it becomes more and more, easy to manage. So I think definitely it just takes time. Your body will adapt, and so
yeah.
Cool. I have one more question, then we can start to wrap this up. So we talked a lot about, the food and how it affects cognitive impairment. Do you think any sort of, in the future, obviously, is there any way that maybe our diets could be affecting diseases like Alzheimer's or even, dementias?
Well, there's definitely some evidence that is growing, linking the excessive use of carbohydrates and impaired blood sugar control. So a dysglycemia kind of situation. So your blood sugar is imbalanced on a regular basis with the development of Alzheimer's. It's a complex type of etiology. So it's not just eating loads of donuts will make you get Alzheimer's, because you could have a very resilient system that allows you to cope with that sugar. So it's not going to have the same effect on everybody. But if the right combination of factors, or the wrong combination of factors, really in this case, is present in your body, then you're more likely to suffer as a result. So if you have something like an ApoE, the ApoE gene that is partly responsible for the development of Alzheimer's, because it carries, it's a fat carrier. Imagine again, another analogy, your arteries a bit like a mine with those trolleys, those little trains with coal in the mine, and and they go into the brain, and that's kind of like a finishing point there for that train line in those mines. And and that train is completely overloaded with coal or with cholesterol in this case, because that's what ApoE does very effectively. If you have the most effective combination of that ApoE gene or variant, which is the ApoE4, if you have two copies of that, two alleles of the ApoE4, or even just one, you're bringing a lot more of that cholesterol into the brain that could be then oxidated, and high glycemia, so high glycemic foods have been associated with higher oxidation. So when you've got that combination of high oxidation and high cholesterol coming into the brain, then there could be a lot more oxidative damage happening in the brain. And that's not good when you have then a propensity to accumulate more misfolded proteins as well, like the beta amyloid and so on. So yes, so definitely the short answer is yes. A more sugary diet or a more carbohydrate intense diet. So anything from the more obvious things like pastries and donuts and sweets and candy, crisps or chips and all of that kind of stuff, to the less obvious, that could be bread and so on. Again, if your diet has got a lot of bread and a lot of potatoes, you could be doing almost the same thing as a little bit of candy and a little bit of chocolate. So there's definitely an interaction between the levels of sugar in your diet and your propensity to develop a neurodegenerative disease along the lines of the Alzheimer's spectrum.
Yeah, wow. Okay, cool. Well, excited to see more research come out on that and see where the future is. Well, thanks for coming on today. You've kind of blown my mind. You know a lot about all this topic. I'm going to have to go back and listen to it a couple times to kind of soak in all the information. So thanks for coming on.
Thank you, Julian. You're too kind.
I would love to have you on again if you're interested. I already am starting to develop
definitely.
questions for you. So we'll have to keep in touch, and good luck with everything. So thanks for coming on.
All right. Thank you so much. Take care. All the best.
Take care. Have good luck writing your paper today. And
thank you. Speak soon. Bye-bye.
Okay. Bye-bye, Miguel. Thanks. Okay, that's it for the Straight from a Scientist podcast. Check us out on iTunes, or we also actually have our Kickstarter up. Our Kickstarter is live till Thursday. We're getting close to our goal. So please, if you enjoy our podcast, support us. And yeah, that's it. That's all, folks.